We're going to have to go without the microphone initially and use the computer microphone. We have some technical problems, but at least people can hear us on Zoom. So anyway, a pleasure to welcome Dr. Dan Yosher, who's chairman of neurosurgery at University of Pennsylvania, who's our Irma and Kalman Bass innovation lecturer. And I've known Dan since 1999 when he came as a epilepsy fellow to work with Nick Barbaro at University of California. He did his training at Methodist and Baylor when they were together, and then went back and worked at Baylor, was chairman of neurosurgery at Baylor for 10 years before he moved to University of Pennsylvania in July of 2020 to assume the chair position. Many of you will know that one of the Nobel laureates for the RNA sequencing work in COVID actually is from the University of Pennsylvania and had an appointment in the Department of Neurosurgery at Penn, correct? Okay, so I think financially, University of Pennsylvania will now be well positioned for the future based on licensing of that technology to pharmaceutical companies. But anyway, Dan and I have always jousted with each other verbally and in all good fun over the years. And it's a pleasure to see the success he's had. And we're looking forward not only to this lecture, but his future accomplishments. Dan, please.
It's a great pleasure to be here today, and I'll quickly dispense with the disclosures. We will be talking about technology, but I really have no financial interests in any of the material discussed today except for a patent. I want to first thank the Bass family for allowing me to be here today, and I want to make the point that they are visionaries in investing particularly in technology and innovation. That is so important. And bringing Mike McDermott to Bass, he's been here for 3 years. Mike is the epitome of restless energy, not accepting the status quo, always trying to do better, always trying to fly higher and faster. Mike is a great teacher. Constantly, he says he's jousting with me. He's always teaching. And as you know, going back to high school, those relationships never changed. Now, I'm a middle-aged man now, but Mike still thinks of me as his fellow, back then, and I accept that. One of my favorite memories of Dr. McDermott is I was at a neurosurgery meeting with him in Florida, not far from Pensacola, and we're all the fascinating presentations. And at some point towards the end, Mike taps me on the shoulder, whispers to me, "Meet me in the lobby in 5 minutes." Like, okay, yes, sir. So 5 minutes later, I go to the lobby, have no idea what's going to happen. And Mike says, gets in his rental car. We drive 15 minutes. He takes me to the Air Force Museum of Aviation Technology in Pensacola, Florida, near the Air Force Base. And we looked at all the magnificent feats of engineering, including this SR-71 Blackbird. And Mike, always the teacher, explained to me that this plane is the fastest publicly acknowledged, highest flying, fastest plane ever produced, 3 times the speed of sound. It was engineered to fly high and fast, and when it was on the ground, it leaked oil because the joints would expand at altitude. And this kind of innovation is the spirit of what we want to talk about today. Restless energy to pursue new, better, different, never knowing where it will take you. And, yes, I am proud to come from the University of Pennsylvania, where we are also pursuing this restless energy and this spirit of getting better in recognition of our founder, Benjamin Franklin. You can see there on the right is the Old World Penn Hospital University campus, and on the left is the New World, brand new building, the new pavilion. And this is where you stole my thunder. And I want to highlight the Bass family's commitment to philanthropy at this wonderful health system and institution, and their interest in innovation and technology. Katie Carrico there on the left side of that picture was a relatively unknown, frankly struggling researcher in the Department of Neurosurgery at the University of Pennsylvania that I chair today, and struggled to get grants. And you'll hear about this on the news, of course, because last week she won the Nobel Prize. And I think this is a lesson that without people like the Basses supporting this kind of work, you never know where restless energy, creativity, innovation, pursuit of knowledge based on intellectual curiosity. I want to understand how fast jets fly. You never know how that's going to affect how we do brain surgery. And in this case, her work in neurosurgery, she was actually researching using technologies, developing new technologies for stroke, but you never know where things go. And she ended up inventing the mRNA technology along with her colleague, Drew Weissman, that subsequently led to the development of both the Pfizer and Moderna vaccines that have saved millions of lives across the world. So I want to salute your health system, your medical school, and most notably your philanthropic supporters for allowing this kind of energy to continue. And I hope that their investment will produce similar rewards. You never know who it'll be and when it will happen. So in that spirit, I want to talk a bit about the work I've been focused on for the last 20 years. And it is work that is based on, again, two motivations. One, I'm curious, just as Mike is curious about how SR-71s work and how race cars work, I'm curious about how the brain processes visual information. That's one motivation, intellectual curiosity. The other motivation is, can we use this knowledge to help people who right now have no other option? So going back in history, one of the fathers of neurosurgery was Wilder Penfield, who worked at the Montreal Neurological Institute, founded the Montreal Neurological Institute. And Penfield was famous as one of the first neurosurgeons to take the concept that you can stimulate a spot in the brain and either turn on the neurons in that area or turn them off. Either replicate the function of that spot in the brain. For example, if you stimulate the part of the brain that controls the right arm will twitch. The right hand will twitch if you're in the hand spot. The shoulder will twitch if you're on the shoulder spot. The leg will twitch if you're on the leg spot. And he would do this in awake surgeries in patients with epilepsy. And of course, he was doing it in order to avoid hurting the critical parts of the brain when he would take out the area where the seizures were coming from. But he also learned a great deal about how the brain works. And really, this work should have won a Nobel Prize. It never did, but it should have. And one of the things he noted, people mostly focused on, I'm showing here the famous picture of this sort of homunculus, this enlarged body, because the face has more representation and the hand has more representation than your torso or your leg. That's for motor and sensory function. But if you look here, he also explored the visual part of the brain on occasion. And what he noted was that when you stimulated the back of the brain, let's say you stimulated the back of the right side of the brain, patients would see a spot of light in the left side of their visual world. And he deduced that there was a map of the visual world in the back of your brain, and that the information that was transmitted onto the retina, when you all are looking at me right now, that image is actually really being processed in your brain. What I would say is your brain does most of your seeing, not your eyes. Your eyes do a lot, of course, and each step is critical. But ultimately, the processing of information in the visual world is in your brain. And you may be surprised to know, but more than 30% of your brain is strictly devoted to processing visual information. That's how important vision is. So, like Dr. McDermott, I'm a neurosurgeon, and part of my job, or the essence of my job, frankly, as a neurosurgeon, is to take care of patients. And I've had the privilege of taking care of many, many patients with epilepsy and brain tumors causing epilepsy. And in many of these patients, in order to understand their epilepsy better, we have to put electrodes in their brains. And I'm sure I know you have an active epilepsy center here and you do this kind of work. And we've used that opportunity over the past 20 years to study how the brain processes visual information. As I said, mostly because we want to understand this, driven by intellectual curiosity and an interest in innovating. And what we've done is, and I show you this in this figure here, we've been able to map receptive fields. What that means is, and I think I have a slide that illustrates this coming up, is we're able to record from a spot in the visual brain. And as the patient is sitting there and we're recording, we project images of checkerboards. They're very salient. They light up the brain like crazy. We put checkerboards in different spots on the screen. And we can tell from our electrode which area in the visual world activates a specific spot in the visual brain. And what we've been able to show, going back to 2007, is, and some of this work, of course, had been done in cats and monkeys previously, but it had never been done in humans, certainly on a physiological level. And we were able to show the precise nature of these receptive field maps in the brain. And the take-home point I want to make here is, and here's an example of it, each square here represents an area in the visual world. And you can see when we flash a checkerboard in this area in the visual world, the spot in the visual brain that we're recording doesn't really get activated. Here, it doesn't really get activated. Here, it doesn't really get activated. But you can see in this area on the map, all of a sudden, look at that burst of activity. And that corresponds using mathematical transformations and Gaussians fit to a 2D Gaussian. I won't get into the boring math parts of this here, but basically this shows this is the receptive field. The spot of the brain we're recording from in this experiment only lights up when this part of the visual world is activated. So basically, the lesson I want you to get from this example is the visual brain has very specific spatial maps. A spot in the right visual brain is tied directly to a specific spot in the left visual world. If I put a stimulus, a visual object in that spot in the left visual world, this spot in my brain is being activated, and it's a point-by-point map. The next thing I want to tell you is there are lots of maps in the visual brain. It's not one map. There are probably 20 to 30 repeated maps in the brain going from the back of the brain to the front of the brain. And those maps are all different maps. Imagine if you look at your Gaia app, you have topographic maps, you have satellite maps. The maps in the back of the brain, the early part of visual cortex, are very spatial. It's a point-to-point veridical representation. But as you get into the later areas of visual cortex where the information is processed more, the maps become specialized for form or function. For example, there's a part of the brain called area MT, which maps out movement in the visual world when dots are moving. There's a part of the brain in the ventral temporal cortex, and that's the example I show here, that responds to faces, the fusiform face area, or to places, the parahippocampal place area. Our brain is highly evolved to recognize forms and visual information. We do this without even thinking about it every day, but imagine the complex transformations of information that is taking place in the world's greatest computer every time we just effortlessly look at our friends, at our family's faces, and recognize them instantly, and without even having to work very hard at it. That's all happening because we have all these maps in our brains. And one of the things we did in our lab is work on recording from these advanced, more complex, more specialized maps later in the hierarchy of visual processing. And here's an example, as I showed you, of this is a spot in the brain right here, and red represents how it responds to faces, in this case, a picture of Abraham Lincoln, where you can see it doesn't respond very much to a picture of a house, even though these have the same amount of black and white voxels in them. So it's not that the brain is responding just to blurs of black and white, it's responding to the actual face versus a place. So I talked about how the brain responds to natural visual stimuli, meaning information that's directed to the brain through the eyes, through the retina. I mentioned before that Dr. Penfield, the famous neurosurgeon in Canada, did that work where we would stimulate the brain and reproduce function. So in our laboratory, working on patients largely in the epilepsy population that we serve, we also did experiments stimulating the brain, being very careful, of course, because when you stimulate the brain, you don't want to stimulate too much, heaven forbid, and produce a seizure. But the good news is these patients are all being studied for epilepsy, and they're in a lab where they're constantly being monitored, their brain waves. So we were always able, as we stimulated, to make sure we weren't stimulating at a level that would produce a seizure. And we really would almost never, I'd say essentially never produce a seizure with this kind of research stimulation, because we always stayed well below the level that would be potentially dangerous for the patient. And of course, we're grateful for these volunteers. Let me make it clear, all these patients were in the hospital and had electrodes in their brain for their own clinical reasons. They weren't in the hospital to do this research, but they're sitting there in the epilepsy monitoring unit. And I think I'm visiting your unit today. I think it's on my schedule. So they're sitting there waiting to have seizures so their doctors can understand where their epilepsy is coming from. While they're waiting, we ask them, would you like to help us and participate in this research and help us understand the brain better? And many, if not most of the patients say yes. They're sitting there, and most people want to help. And we're very grateful to their cooperation over the years. So when we stimulate, as I said, the back of the brain, we produce a perception of a blob of light in a corresponding location on the opposite side of the world. And that's what this cartoon is supposed to show here. So one of the questions we asked in our research is, remember I told you there's a lot of visual areas in the brain, and some of them are specialized just for blobs of light and spatial locations. And some of them are specialized for forms, for movement, for faces, for shapes. We want to know, using the crude electrical stimulation that we can use as doctors right now, what would we see when we stimulated a specific area in the map? And on the bottom in the x-axis there, you see the names of a variety of visual areas. From left to right, it goes from the early part of the visual processing to the late part. Remember, I told you the early part, the maps are fairly simple and spatial. The late stages, the maps are much more complicated and abstract. It's like art, by the way. And this is why abstract art, like Picasso or Mondrian, tickles our brains. That's why people like abstract art, because their brains play with those forms and process them and deconstruct them in the various maps that exist in our brains. So we were interested in knowing, can we we knew we could activate the very back of the brain. V1 is, as you see there on the bottom left axis. We knew that would work. We studied dozens of maps in the brain. And what we found, and we published this in PNAS in 2009, was that the only areas where we could reliably stimulate the brain and produce a clear perception in the visual space was in the relatively early areas, not in the later areas. And that's kind of interesting. Why would that be? Because we know those later areas are very involved in vision. I mean, we know that if you take out the bilateral face recognition areas and this is, been done in animal models. Obviously, we'd never do this in people. But if you take out these bilateral, and this is one of the reasons we never do bilateral resections in the temporal lobe, you destroy a person's ability or a primate's ability to process complex visual forms. They become face blind, for example. Very rare, because you rarely have damage to these two sites on both sides of the brain. But we wanted to understand a little better why we couldn't stimulate those areas and produce. Patients don't say, oh, I see a face. And this is probably the simplest explanation. In the early parts of the visual brain, which I show on the left, V1 is the earliest part. V stands for vision, and 1, it's the first stage of the visual maps. It's a very simple map. It's a spatial map, and adjacent areas are in the visual world are represented in adjacent areas in the cortex. So when you stimulate crudely, you bang on the piano, so to speak, you get a reasonable sound. You get activation of a contiguous area in the visual map, and the brain is able to process that and interpret it. It's different than how that brain is normally activated through the eyes, but it's close enough that the brain can understand it. But when you go to the later visual areas, and that's a famous map from the monkey inferior temporal cortex showing how the organization of body parts and faces is in that ventral temporal lobe, that's a much more complicated representation. And when we stimulate the brain using we're not as good as what God or evolution produced. We're stimulating the brain with an electrical current. And when we bang on the piano, we're producing something that probably doesn't sound like music to the brain. The brain doesn't understand it. And that's probably why it doesn't produce a percept. Someday we may be able to bang on the piano more elegantly and produce a useful signal in that stage. But right now we're really only able to do it in the early stages of visual cortex. I'll briefly mention, this is some of the work that I'm most proud of. It's probably not relevant to what I'm going to talk about later on, but we also studied how the brain takes information from the outside world and puts it in conscious perception. So you all should recognize that your brain is being bombarded constantly by visual information. And most of it, you're not even aware of. You ignore it until you pay attention. You can do there are all sorts of psychophysical tasks where you can look at a picture, they'll flash a picture in front of you, and they'll ask you, was there a gorilla in the corner? And you don't notice it until you're told there's a gorilla there, and then you can't not notice it. That's because our brains are designed to be very selective in the information they process. It wouldn't be useful if, as I'm talking to this audience, I was noticing the pattern in the carpet. It would distract me. And the brain has evolved to be selective. So we found that, where does that selectivity come from? We found that if a visual stimulus does not activate this area of the brain called the temporal parietal junction, or TPJ, even though the visual information entered the visual map, it won't enter your conscious awareness. And this probably relates to the clinical phenomena. We may have some neurologists and neurosurgeons in the audience of inattention. When you have a high parietal lesion and the patients they see, but they can't attend to visual information, inattentional blindness. So I think it's important to note, and I'll just jump ahead from here, that even when information reaches the brain, there are other steps that have to happen before visual information enters conscious perception. So we also looked very carefully at how activation of the back part of the brain. Remember, I told you the back part of the brain, we were very able to produce those percepts of spots of light. How precise is that activation? Can we precisely predict when we stimulate the back of the brain, V1, the occipital cortex, where the visual stimulus will be perceived? And we did a series of experiments over a number of years and developed a mathematical model. And the short answer is we are very good. If you show me a brain and show me a spot in the brain, I can tell you with a high degree of accuracy where the spot will be perceived when you stimulate that part of the brain, how big the blob of light will be based on these mathematical computations and our understanding of how visual cortex works. And that's going to be important if we want to use this latent ability to produce visual percepts in the brain by stimulating the brain. If we want to use it to help blind people, that'll be very important. We also looked at stimulating more than one electrode at a time. And think about it, if you want to use this someday to restore vision to patients, patients don't want to see a single blob of light. They want to see forms, patterns. And in order to produce patterns, and I'll talk about this shortly, you need to combine more than one blob. Like your TV is made of a 1080 by 720 blobs of light. That's your pixels. That's a low resolution TV. You guys probably all have 4K or 8K TVs now. So we did a lot of work looking at how you stimulate more than one electrode at a time, stimulate 3 electrodes at a time. Can patients reliably identify the patterns of dots that they see when you stimulate at the same time? And it's pretty challenging, actually, because remember, the brain is accustomed to being turned on by visual information entering the retina. We're doing this in a very artificial way. And I keep bringing that piano analogy because I think it's a very useful one. You can play beautiful music with a piano if you stimulate the keys naturally. But if you're just using your elbows to play the piano, it doesn't really sound like music. And that's the challenge we have, is by stimulating the brain, we're activating that visual those visual neurons in a rather crude and elegant way. Here's an example of 4 to 5 electrodes at a time. So let me jump ahead with that segue to what we really want to talk about today. So most of what I've discussed is fascinating from an exploratory standpoint, and we're all intellectually curious, and we want to understand how the brain works. We want to understand how mRNA works, drawing from the analogy of the recent Nobel Prize at Penn. But it's important to understand how mRNA works, but we really get excited when we can use mRNA technology to save lives. It's important to understand how the visual brain works, but it would be really great if we could also use this understanding to restore vision to people who right now have no hope. And that is the basis for developing what I call a VCP. That stands for visual cortical prosthetic. Visual, vision, cortical, that's the part of the brain, the outer layer of the brain, the cortex, and a prosthetic, of course, is an artificial device. And people have been attracted ever since Penfield described in the 50s, stimulating the brain, producing artificial images of a spot of light. It makes a lot of sense. If you stimulate one electrode and produce one blob of light, why don't you stimulate a lot of electrodes, knowing that there are maps in the brain, and use those spots of light like pixels on a screen and produce forms like an image of a leaf? That's the tantalizing prospect. We have visual maps in the brain. We can turn on one spot in the map at a time. Can we turn on a lot of spots in those maps at the same time and create an artificial image of a form, bypassing the broken eyes in blind patients and putting that information directly in the visual brain? And that's what we would anticipate, a contour of a leaf in this case. So actually, people have done this for a long time. And I sometimes show a Star Trek video when I give this presentation. I don't think Miami is a nerdy enough town. You know, this is a cool city. You guys don't watch Star Trek. But some of you may remember there was a character in one of the Star Trek shows that was a blind officer in the Starfleet who had some device, wore a visor, and could see the visual world. That's based on this work. The guy who wrote Star Trek, Gene Roddenberry, knew about this research that was going on in the 60s and 70s. Now, it was very crude. This was in England, in this paper Brindley and Lewin published in 1968. The work was done in the mid-60s. You can see the computer filled the room. And the patient, is incredible work for its day. They actually were able, they had hardware inserted in the brain. This was all, remarkable. And they were able to get a few spots of light. Obviously, the patient, the blind woman here, could not leave the room. All the device was harnessed to the room. But it was incredible for its day that they actually were able to do this. The person who really famously championed this in the 70s and even into the 80s was a neuro engineer and inventor at Columbia University in New York named William Dobell. And he developed a portable version. You can see the blind patient walking around with a little handheld computer. Now, the concept in all these devices, I should make this clear, is the patient wears glasses that have a camera on it. The camera, and you can see it there in the bottom right image. The camera captures the information that the patient would be seeing if they could see. That information is converted into a visual pattern by a little computer. In this case, he's carrying it on his knapsack at his side. The computer then converts the visual information into a pattern of electrical stimulation in the brain, trying to turn that into dots that can turn into an image that a patient can use to see, to behave, to navigate. The technology, of course, was crude, and Dobell was famous for, he was a strong self-promoter, let's put it that way. He was on 60 Minutes and the New York Times and Time Magazine. The best he ever really did was he had a paper in Nature in 1976 where he showed that he could train the patients in what he called visual braille. But were those patients really seeing letters? No, what he taught them to do is he would stimulate a series of dots. So all they saw was a few bright dots in their visual brain. And he taught them to convert those dots. This pattern equals letter A. This pattern equals letter B. This pattern equals letter C. So they weren't seeing A's and B's and C's. They were seeing dots that they learned to convert as a code into letters. Now, it was impressive. It was big deal. But it's not really what we want. We want to develop a prosthetic where patients can actually see forms, because the world is not limited to 26 letters. I want to be able to recognize you and you and you. I want to be able to navigate in an unfamiliar hallway. So I mentioned one other trial that took place in the late 80s, really, and wasn't published until 1996, was at the NIH. My old boss, Bob Grossman, remembers this well. Don Quest worked with Dobell when he was a medical student. This was a trial. The NIH was really excited about this work, and they implanted a subject with penetrating electrodes, smaller electrodes, thinking they could stimulate more finely, have better resolution, little tiny dots. And that trial, unfortunately, didn't go very well, and there were complications. It becomes a little more dangerous when you actually are penetrating the cortex as opposed to putting electrodes right on the surface. And I would say from the 1990s till about 2010, 2015, the field pretty much stopped. People gave up for a while. The technology wasn't good enough. And then all of a sudden, it exploded. Right now, there are 3 ongoing clinical trials trying to do this kind of work in human patients. Why was there an explosion? Well, think about it. Mike, hold up your iPhone, please, as a prop. So that iPhone is thousands of times more powerful than that room of computers I showed from the 1960s. Engineering has advanced incredibly, and the concept of doing this is much more approachable now. We have pretty good hardware. We certainly have the computers to rapidly process visual information captured by a camera and turn it into a useful pattern of stimulation. The question is, can we interface with the brain well enough with our electrodes? And do we use a code of entry? When we bang the piano, are we banging it accurately enough to produce music, or are we just producing noise? And that's what I'll talk about in the next few minutes. So the 3 trials I'll talk about, one is the Orion Second Sight trial, which I am one of the investigators on, which is still ongoing. Another is the Cortivis trial, and I just met my colleague, Dr. Fernandez. This is from Spain, and they are working with microarrays using a Utah array. And the third, which is ongoing, is the ICVP, or intracortical visual prosthetic slide project, and that's a team led by the Illinois Institute of Technology and neurosurgeons at Rush University. And I'll talk about each one a little bit. So first of all, the Orion trial, which is the one I worked on, did not try to put in thousands or hundreds of electrodes. They're just 60 electrodes, not that many spots. The device goes on the interhemispheric portion of the brain. There's a wireless computer transmitter. So that's the nice thing. There's no wires coming out of the patient's head. Anytime you have wires coming out of the brain, ultimately there will be problems. So it's much nicer to have a totally wireless device. The patient can walk around with an almost invisible scar. And that's one of my patients there, blind patients that I implanted. And this trial took place at UCLA and Baylor College of Medicine initially. And of course, I moved from Baylor to Penn, and that site shifted to Penn. And we have implanted, together with my colleague, Nadar Paratian, who actually was at UCLA at the time and has since moved to the University of Texas Southwestern. We've implanted 6 subjects and worked with this company to determine the usefulness of this product in producing visual information for patients. So I want to show you some examples here. This is one of our subjects. Let's sound on. So he's using the prosthetic to find where the square is on the screen. You see how he's scanning around with this camera. This patient has no light perception. His pupils do not react to light.
Correct.
Hasn't seen in 27 years. That shows what the computer is doing, by the way. It's looking at the information captured from the visually seen
Correct.
and turning it into a pattern of dots. Now, as a control, we test it with the device on.
Higher.
He's just
Right.
guessing now. He has no idea. And then we try to test it in terms of activities of daily living. And you see, it's not easy. He has to scan around with the device. He's using it largely as a contrast detector. You see how he methodically scans. Just remember, this is an early feasibility. But here he is navigating the hallways outside my laboratory. Is he seeing the objects in there? No, he's seeing brightness and darkness. It's helping him navigate the corridors. So I admit that was a moving moment for me. He says, I definitely see the sun, and you can see where his shadow is behind him. He is seeing the sun. So, and that was moving for him too. And, you know, it does show that there is capability here. There's no question he's getting visual information that he otherwise would not be receiving. But remember, to really get where we want to go, we want to be able to see forms, objects, faces, letters. And with the device that we've been testing, and we haven't published the final results on it, but I will tell you the preliminary results are it is effective in identifying bright blobs in a scene. It functions as a useful light detector. Patients can perform behavioral tasks, but in order to make patients submit to having this implanted outside of an experimental trial, we probably need to do better. And I think it's an important first step. And I want to be careful here. I have a lot of respect for my colleagues in this company who developed the technology, the engineers and the scientists. I think they're all intellectually honest and good people. I don't have any financial interest in this, but I am proud to have participated and continue to participate in it. And I should note, this is also funded by the NIH, by the National Institutes of Health. We still have a ways to go, but we showed some feasibility. And here, just this is a graphical demonstration of what I showed you before, how the patients do far better in these performances. When they don't have the device on, they're just guessing. They do no better than chance. When they have the device on, they perform quite well at the task. So one question is, remember, in this study, we use these large surface electrodes. They weren't penetrating the brain. What if we use arrays like this, which have tiny little pins that penetrate the brain? Or maybe someday a surface electrode with tiny little electrodes and thousands of them. Would it be better? Is the problem that our electrodes are too big and too few in our trial? Very possible. So our colleagues at the Universidad Miguel Hernandez, and that's led by Eduardo Fernandez from Spain, have published a number of papers. They have implanted their second patient, and they've reported on their first patient. And I just met with them in Germany at a symposium just a couple of weeks ago. And they collaborate with an outstanding scientist in the Netherlands, visual neuroscientist named Pieter Wolfsema. And they've used a off-the-shelf product called the Utah or BlackRock microarray, which many brain-computer interface investigators have used across the country, usually for motor prosthetics or language prosthetics. Like Jamie Henderson uses it in his language studies at Stanford, for example. So they use this device. This is not wireless. There's a wire coming out, and these are good, again, good ethical scientists and physicians. So they are only implanting their patients for 6 months because there's a wire coming out. They have blind volunteers, and they're capped at 6 months, because you don't want to leave a wired device coming out of your head indefinitely. So this is not trying to test a device that will be implanted long-term. This is a proof of concept. Did the microelectrodes work much better than our larger surface electrodes? And the short answer is not really. In fact, the maps that they produced were not as orderly. We were able to show very clear, orderly maps. Their maps were not as orderly as ours. Sometimes patients saw shapes or percepts they described as O's. or lines, but sometimes the percepts didn't match up or didn't fuse properly. It was complicated. They're doing a second subject now and they're hoping for better. And I'm certainly rooting for them. They're a good group. The other good group is this group based in Chicago. And these slides are courtesy of Dr. Danieli and Dr. Troik, who are the leaders of this investigation. Troik is an engineer at the Illinois Institute of Technology. Their concept is they want to use the pin-like electrodes, the pin cushion electrodes, but they have what they call a floating tile, wireless floating microelectrode array. And they've implanted one subject, I believe. Maybe they just did a second one. I'm not positive about that, but they've only reported early results on one subject. Yeah, here it is. The first surgery, and this was done by our colleague Rich Byrne, did the surgery. And this was in February of 2022. And they inserted 25 of these floating microarrays. And from an engineering perspective, the device is working well. It's wireless and it produces electrical currents. The only results they've really presented is, this is from something they just presented. And again, I thank them for sharing their slides with me. I, of course, have their permission to use these. The results that they present show that they do get maps in the visual world. They have not presented any data on how the patients perform. Are the patients able to detect forms? Are they able to navigate? We don't know yet. We certainly have high hopes. But it is not yet clear that we're able to produce useful visual forms. Why is that? I think it's the same story I described before, where when you stimulate later visual areas with crude stimulation, it's hard to get an interpretable percept. I think when we unnaturally activate multiple spots in V1, it's hard for the brain to take that unnatural integration, the unnatural activation of multiple spots and turn them into a unified form. That doesn't happen easily. And that's the last part of the story I want to talk about. I think I'm nearing the end of time, so I'll avoid some of these. So we worked on this for years, and it was very hard to get patients to combine the phosphines. That's what those spots of light are called, phosphines. The goal is to use phosphines like pixels on a screen as combinatorial elements that combine together into a useful form. It's not easy to do it by stimulating all the pixels at the same time and thinking that will become a shape. So we chose a different tactic. And for any of the neurologists in the audience are certainly familiar with one of the best tests of somatosensory function in patients is we draw letters in someone's hand where there's a very detailed spatial map in the brain of your hand. And if you draw the letter Z in your hand, try it. It's very easy to tell that this is the letter Z. On the other hand, if you took a Z and pressed all those spots at the same time in your hand, it's much harder for your brain to process that information. So he said, why don't we try this in our patients, in their brains? And this was not part of the Orion study. This was a side project that my laboratory did. And we published this in Cell a couple of years ago. And the idea was not only would we skywrite, you guys have all seen skywriting, skywrite the images of letters in the brain, but we would also create virtual electrodes. Remember, I said we only had 60 electrodes. So we wanted more coverage. So to create a virtual electrode, we would stimulate using a technique called current steering. We would drive the current in between the 2 points, basically creating a virtual electrode between the 2 physical electrodes and sweep across the brain. And this is sort of the idea of what a patient might see. This would be concurrent stimulation of the letter Z. Not so easy to turn that into a Z, is it? And this would be dynamic stimulation of the letter Z. It might be easier. So we tried this initially in our epilepsy patients. And this was a patient who only had a small number of electrodes. I think to do this study, we only had 6 electrodes, yet we were able, if we stimulated those electrodes concurrently, they just saw meaningless blobs. But if we stimulated them dynamically, and look what the patient saw. This is a sighted patient, not a blind patient. And this was an aha moment. This is the first time we were able to produce a useful, this patient had no training, had no idea what we were showing, said just draw what you see. And we did this in this number of other patients. This is a patient that had, again, a sighted patient, not a blind patient, patient with epilepsy where we had a experimental array in the visual cortex, and they could see multiple letters and did very well in performing distinguishing between these letters. Then we moved to one of the preliminary blind patients. This is the first blind patient we tested. This patient was not implanted with an Orion. This is before the Orion had FDA approval. This patient was implanted with a NeuroPace, a device developed for epilepsy. We got special permission from the FDA in one subject to test this as a precursor to doing the trial in blind subjects with the full Orion. And this courageous young woman who's blind volunteered for this. When we use the electrodes all at the same time, we got nothing. Now watch when we're stimulating them in sequence. There she draws an upside down view, which is what we desired. And by the way, why do we have that dot on the screen? It's very important for patients to fixate or direct their eyes and their attention to one spot. Because when you move your eyes around, that actually shifts the frame of reference and all the voxels are offset. Think about it. So you're constantly messing up your image. How do you get a blind person to concentrate on a spot on the screen? With the sighted subjects, we were used to that. We would just put a cross on the screen, they would stare at it, and then they would do the task. So in Texas, we call this a nubbin. We would put a little raised dot on the screen and have them touch it and say, okay, and we know you're blind, focus your attention on that dot. And that inhibited the eye movements that might get in the way. So this was exciting to us. With a few electrodes, we could do this. And then with the Orion device, and I already showed you with the Orion device, the impressive, albeit limited value or utility of it. It was useful as a light detector. Remember, the patient would scan around and would be able to see the objects on the desk, on the screen, but they couldn't see forms or shapes or letters. We took the same Orion device, and instead of stimulating all the electrodes in the same time and assuming that they would combine into a form like voxels on the screen, we stimulated them dynamically. And this was with no training, no effort. Yeah. He is, of course, blind. And he's identifying one at a time. We simulate the letters.
Now.
Draws what he sees.
Sure. Starting now.
So using a limited number of electrodes and dynamic stimulation, we could generate forms that the patients could readily identify. And this is something that no one had been able to do before and does not necessarily immediately equate into an off-the-shelf device that we're ready to implant in patients. But this is like walking on the moon as a step towards colonizing the moon. It shows that it is feasible, that we can inject information in the brain in a blind patient and have them see a letter. And I think it is an important early feasibility evidence for what we can do. And here's just some of the data supporting this. And I'll just jump ahead. So my last slide, where do we stand right now? I mentioned there's 3 of these trials ongoing. The electrodes seem to be a limiting factor right now. I didn't even try the penetrating electrodes because I didn't think they were ready for prime time yet. And one of the problems with the penetrating electrodes is over time, they seem to damage the brain a little bit. And, you know, there's no harm, thank God, to these blind patients because this is a visual part of the brain. I don't want to imply that anyone's being harmed. The question is, though, nobody wants a device that works for 6 months. They want a device that lasts. And the electrode technologies are probably not good enough yet. We want penetrating electrodes because they're much finer and they use lower currents. There's a lot of work now in developing flexible electrodes, or one of the amazing technologies the engineers are developing are electrodes that are rigid when they go in so they can enter the brain, and over a few weeks they become soft and pliable. And people are developing these things right now. So the engineering needs to get better. Visual field coverage needs to get better. A lot of, I didn't say this before, a lot of the visual brain is buried in the sulci, in the little nooks and crannies of the brain. We have to think about how we can access those areas. We have to have better phosphine maps and fitting. And I think really important, and this is what I wanted to emphasize, that as I described our rather I would say, innovative use of dynamic current steering. It's probably not going to be just dumping all the information in the brain is not going to be the most effective way. We have to, and the way I would look at it, we need better hardware. The hardware, of course, is the electrodes, and we need better software, how we activate the brain. But I hope you see from this talk today that there is real hope here and real potential here in our lifetimes to see this develop as an off-the-shelf technology that will restore vision to the really millions of patients across the world who right now have no other options. So again, I want to, well, first let me acknowledge, my, outstanding partners and collaborators, and of course the patients, courageous patients and family who have volunteered. I want to thank the funding agencies, the NIANDS, the National Eye Institute, and DARPA. And I want to thank Dr. McDermott and the Bass family. And the last point I want to make is, without people like the Basses, we can't fuel this kind of technology and innovation. And I think we want to fly 3 times the speed of sound. Now we want to fly 10 times the speed of sound. And this kind of restless energy, I think, will take us forward. Thank you very much.
Thanks, Dan. I think all of us would agree it's pretty impressive. And apart from the visual prosthesis, there are other engineering moves, and neurosurgeons primarily lead those. One of them is artificial speech, Eddie Chang, and then the Italian group with cortical stimulation to promote ambulation. Do you see other areas where we might be developing prostheses for other neurologic function?
Well,
come over here, because the
thank
mic's
you.
here.
The area where I really want to draw a key distinction there is what Dr. Chang, for example, and Dr. Henderson are the 2 national leaders, 2 outstanding neurosurgeon scientists. I should add that Mike trained one of them, and he will openly and acknowledge that Mike trained him. That's Eddie Chang, who's one of the preeminent people in the world in this field. He's developing a speech prosthetic, basically a device that for people whose brains, for whatever reason, are unable to produce speech, they have ALS, they can't move their lips properly. Those devices are read out devices. So what a speech prosthetic does is it takes the signals from your brain when you're trying to speak, but you can't control your lips, and it interprets those signals. What are the words you want it to say? And that's very challenging. But what we're trying to do with vision is even more challenging. We're trying to write in. We're not just trying to capture information and decipher it and then convert it into a code that spits out words or makes the arm move. What we're trying to do is write in the brain. They're both challenging. I would argue writing in is even more challenging. But I think with these readout technologies, yes, there's a lot that's going to happen. And, paralyzed subject can already control a cursor fairly well just using their brain signals recorded from electrodes, or turn those brain signals into words. And that's, fantastic work. We're trying to write in the brain, which may end up not involving electrical stimulation. It may involve optogenetics. I'm agnostic to how we write in. I just want to make sure we activate the brain in a useful way.
One of the things I've had some indirect associations with visual function through my tumor work with tuberculum meningiomas, and realizing that we could recover patients' vision who were suffering probably from small vessel vasospasm. Intraventricular meningiomas in the region of the atrium, where we had transient ischemia of the lateral geniculate. We had 2 patients we reported, and the blood supply to that region was not well known, but it was to caution surgeons that when they're removing tumors in that area, they need to take the vessels supplying the tumor as close as possible to the surface of the tumor, so they don't interrupt vessels supplying part of the brain. But the real curious phenomenon was with bilateral occipital supra and infratentorial craniotomy for giant falcotentorial meningiomas, where we had the clinical experience in the first few patients. Then I realized something called Anton syndrome. and it's transient in all the patients. And what happens, because we manipulate the occipital lobes bilaterally at separate times, not continuously, the patients would wake up, and for the first 3 days, they were not able to percept, to use Dan's term. They were getting all the retinal information, but they couldn't encode it to realize that they were seeing a face or a person standing in front of them. And the remarkable thing is that they confabulate, they make up stories to cover their visual deficit. Do you have any comments on that
It
syndrome?
just shows they're not
Here,
making
over
up
here.
stories. Part of their brain is getting the information, so they are getting something, but
Yeah.
no, I mean, it just emphasizes the point that so much of what we think of as vision has less to do with your eyes and much more to do with your brain. And yeah, I mean, there are fascinating syndromes where you can manipulate areas and cause face blindness or
Yeah.
inattentional blindness. The brain, the visual brain is incredibly complex. There was a Nobel Prize already awarded for understanding of visual cortex, Ubel and Weasel in the 1970s. And we're still just scratching the surface of understanding it. The offshoots of a lot of this research, I like the theme today is when you do research, you never know where it's going to go. So much of face recognition software is based on people studying these areas of the brain. It's turned into an engineering industry that's affected security, affected how you turn on your iPhone. I mean, it is remarkable. And these software algorithms are just copying what the brain does infinitely better than every software.
Questions from the audience? I have one more question and then we'll conclude. Dan, what's the role now of resting state functional MR and connectomics in evaluating vision?
I mean, it will show you the crude pathways. I mean, it has value and may be useful as we do surgical planning. I know you're very interested in brain mapping for tumors, and that certainly helps people. But it's limited. I think the much more robust activation is when you actually, as opposed to resting state, when you do a visual task, when you actually present a stimulus, it's just much more powerful. But what the resting state does is it shows the topography of the fibers without a question, but you know where they are. I mean, that's the other thing is, a master surgeon, deals with this all the time and knows what area to avoid.
No, I still need image guided help, so trust me. Anyway, thanks to Dr. Yosh for coming in an inspiring lecture. No, absolutely. That was great.